Hi A&P1 students. In this video we are going to continue with the nervous tissue that's in chapter 12, talking about the anatomy, just a little bit more of anatomy that we didn't have time to cover last week. And then we'll go into the physiology. So we're going to focus now on strictly the neurons. So we learned that anatomy from last week, cell bodies and dendrites and axon terminals, also called synaptic bulbs. So now we're going to focus on the plasma membrane of a neuron, and we're going to have to know some specific anatomy about that plasma membrane and the membrane proteins that it contains in order to understand the physiology of a neuron. All right, so realize that the overall function for the neurons is to send, we call them nerve impulses in general, we'll call them nerve impulses, send the nerve impulses down their length and then communicate with another cell, whether it's another neuron or it's a muscle cell or another cell of the body like a gland cell. So that's the overall function of these neurons. So in order for our nervous system to do what it's supposed to, which is communicate and regulate everything, a lot of functions in the body, these neurons have to be able to send a nerve impulse down their length. And so this nerve impulse that we talk about, it's actually something, it's an electrical event, and it actually happens along the plasma membrane of the neurons. So it's kind of events, particular things going on along the plasma membrane. So that's the area that we want to focus on and be very knowledgeable about the parts of the plasma membrane. So we already kind of know the basics, the phospholipid bilayer. We talked about that in chapter 4. So that's definitely there. But we want to focus in on the membrane proteins that are in that kind of embedded in that phospholipid bilayer. And there's specific ones we want to focus on. So we're going to focus on channels and pumps. And so there's going to be different types of them. So that's what we need to be familiar with so that we can understand the physiology of a neuron. So let's go down to, we'll start with the channels. And we got some examples so we can see some pictures showing them embedded in the plasma membrane. So the channels that are going to be embedded in the plasma membrane of our neurons, they can be of 3 different types. So there's 3 different types. Leak channels is one of the types. And then we have ones that are called chemically gated channels. And then we have ones that are called voltage gated channels. So those are the 3 types of channels that you need to be familiar with that are present in the plasma membrane of a neuron. So real quick, remember that channels, when we say channels, remember that these are those channels where they don't require energy, because what happens is the ions that are moving through these channels, they are going down, they move down or with their concentration gradient. So remember, that means they go from an area of high concentration to an area where they're in lower concentration across that plasma membrane. So they don't require energy. So when you hear the word channels, we're talking about the movement of ions down or with their concentration gradient. All right, so the leak channels, these are the ones that the majority of them are open all the time. And that's probably why they call them leak. So ions will move through them all the time, just it's kind of a leaky faucet. You just kind of have ions trickling through them all the time because almost all of them are always open. All right, and they are specific though. So there might be a leak channel for one ion and then a different leak channel for another ion. All right, so those are our leak channels. So you should be familiar with that they're open all the time, and then they're specific for different ions. Then we have chemically gated channels. So these are the ones that are normally closed. And what will happen to cause them to open is a chemical, usually it's a neurotransmitter, will bind to them. So they'll have like a specific area on their surface where there's a chemical that has the right shape that can bind to it. So they're typically closed, but if that chemical's present and it comes along and binds to it, and again, the chemical is usually a neurotransmitter, then they will open. And then when they're open, then the ions then can move through them. But otherwise, if the chemical isn't there, the neurotransmitter isn't there, they won't be open. They'll be closed. All right, so those are chemically gated channels. And then the third type is called a voltage gated channel. So this is the type that, again, normally closed, and the thing that's going to cause it to open is some change in voltage. Now, we haven't quite talked much about voltage yet, but voltage is the definition, and I'll go to our definition real quick because it's on here. Voltage is just the measure of the amount of difference in electrical charge between two areas. So what that means is if we're talking about, let's see if they have a picture. Yeah. So if we're talking about our neuron here, the two areas are outside the neuron, so on this outside of the plasma membrane, and then the other area is on the inside of the plasma membrane or inside the cell. So when there is a difference in that, there's typically a difference in the charges. Usually the outside has a more overall positive charge, just meaning that there's more positive charged particles out here outside the cell than there are negative ones. And then the inside of that plasma membrane of a neuron has a net negative charge, just meaning that there's more negatively charged particles inside the cell and fewer of the positive ones. So that's what it means when we say voltage. So some sort of change in voltage. So anytime, we'll talk about it, there's a voltage that's present when the neuron's at rest, and anytime it changes, anytime that voltage or that difference in the charges across that plasma membrane changes, then we'll get some channels can open that way. And so we call them voltage gated channels. And so once they open, then ions again can diffuse through them, move across the plasma membrane. Okay, so those are our three types of channels that we should be familiar with and be able to describe them. All right, so the next is the pumps. So here are the description of the pumps. They don't have any pictures, but we will look at some pictures later. What you should know about a pump, when they say a pump, it's still a membrane protein that's embedded in the phospholipid bilayer. But what it does, they call it a pump because it's actually an active transport membrane protein. So what it's doing is moving apart some sort of ion, moving it against or up its concentration gradient. So it's moving something, an ion, from an area where it's already in low concentration to an area where it's already in high concentration. So it's going from a low concentration to a high concentration. And that's not the natural tendency for ions. They'd rather move from high concentration to low. So if we need to move ions from an area of low concentration to an area of high concentration, it requires energy. And so there are going to be two pumps that are important when we're talking about the physiology of a neuron. So the first one is a sodium-potassium pump, and we have described that before, and so hopefully that rings a bell. But we talked about that when we talked about active transport in Chapter 4. So that's going to be a pump that's very important in the physiology of a neuron. The other one that's important is a calcium pump. So the calcium pumps are also ones that we'll talk about and why they're important in neurophysiology as well. But realize that they're both going to provide active transport of ions across the plasma membrane of a neuron. All right, so now that we are familiar with the types of pumps and channels that we're going to need to know about as we talk about neurophysiology, I'm going to slide down and we're going to do some anatomy here, because we not only need to understand the difference between pumps and channels, but we need to understand where they're located along the plasma membrane of a neuron. So they have a picture in the textbook that shows a neuron. So we have our dendrites and our cell body with its nucleus and Nissl bodies and neurofibrils. The axon hillock, and then the axon, and this is a myelinated axon, so we got some myelin sheaths around it. And then the axon terminals, or synaptic bulbs. Okay, so this is where more anatomy comes in for the neuron. So I just went over kind of the general anatomy of the neuron. Now we need to kind of focus in on that plasma membrane and remember what type of pumps and channels are present in the plasma membrane of neuron and where along that neuron they are present. And the reason that they're present in certain areas is because of the function of those certain areas of the neuron. So we'll just start with the entire neuron. So dendrite, cell body, and axon, all the way down to the axon terminals. There are pumps and channels that you will find anywhere along the neuron or everywhere along the neuron. So it doesn't matter where, but you will see sodium-potassium pumps along dendrites, along cell body, along the entire axon, all the way down to the axon terminals. So you should know that those are located everywhere along a neuron. The other two membrane proteins are the channels. And remember, these are leak channels, and remember they're specific, so there's a leak channel for just sodium and a leak channel for just potassium. So these are two ions that play a big role in neurophysiology. So we need to be familiar with where their channels are located. So if they're leak channels for sodium or potassium, again, just like the sodium-potassium pump, you can find them along the dendrites, along the cell body, and along the axon, down to the axon terminals. So everywhere. All right, and we'll talk about what role they play when we get to neurophysiology. But first thing you need to just remember is where they're located. So they're located everywhere along the neuron. All right, so when it comes to, so we did leak channels and we did the sodium-potassium pump. So now we're going to look at those other ones that we talked about. So we talked about chemically gated channels. So when it comes to chemically gated channels, where you will find those is in this end. We call it the receptive segment of the neuron because this is the part that receives signals from another neuron. So we call it the receptive end. It's receiving information from another neuron. So in this plate, this is including dendrites and the cell body. So that's the receptive segment of any neuron. It includes the cell body and the dendrites. And if you look closely at the plasma membrane of the dendrites and cell body, obviously you'll see these channels and this pump, but you'll also see chemically gated channels. And there is one for sodium. They say just cation, but we're going to focus on sodium. So there's chemically gated sodium channels. There's chemically gated potassium channels, and there's chemically gated chloride channels. So you will find those in the receptive segment or dendrites and cell body of a neuron. All right, so the next type of channels that we need to know where they're located are the voltage-gated channels. We haven't talked about them yet. So you'll notice that they are only located along they call it the initial segment of the axon, and then the conductive segment, they call this conductive because that's what's going to conduct the nerve impulse to the next neuron. So it's conducting the signal all the way down the axon. That's where you will find the voltage-gated channels. And you'll have voltage-gated sodium channels along here, along the axon, initial segment of the axon, and then along the axon down